Hello and welcome to Lyra Nara. Today marks the start of a 2-video series on cancer. In the first part, in today's video, we're going to talk about all the factors that cause cancer. And in the video that is to follow, we are going to propose a natural treatment plan that really works and addresses all of the problems that we're going to present to you today that have caused cancer in the first place. So please do not forget to subscribe and turn also your notifications on so that you don't miss out on the second part of the series and you get the full picture. Without further ado then, let's get started and learn more about what causes cancer. So cancer goes through different stages. It has different development stages, the so-called initiation phase, the promotion phase with the so-called apoptosis, and the progression phase with the metastatic cascade and the angiogenesis. And it is usually triggered by an unexpected psycho-emotional conflict that alters your biochemistry, but also your cellular metabolism, your cellular membrane potential, and the connective tissue matrix. And the first signs are breakdowns of the capillaries that are mostly visible around the ankles and the nose, and increased HBP and joint as well as gastrointestinal ailments. And the different stages of apoptotic cell death start with cellular shrinkage and chromatin condensation that is concomitant with the formation of membrane blebs. Organelles and the nucleus fragment and the blebs begin a formation of the so-called apoptotic bodies, which are eventually engulfed by macrophages or neighboring cells by so-called endocytosis or phagocytosis. And the lack of release of cellular components to the extracellular fluid then results in the absence of inflammation. Let's talk a little bit more in detail about a cancer initiation phase where everything starts. Because it's important to understand where it all begins, and the human body works as an electrical system, as a battery literally. And thoughts are electrical signals, beliefs, habits and strong emotions are magnetic signals. And those emotions, traumatic events, beliefs, they are all stored in your brain. And when an unattended emotional shock or conflict occurs, there appear in specific regions of the brain, there are edemas appearing in form of concentric rings. And as a result, your autonomous nervous system and your hormonal system will get the wrong signals, and this will in turn disturb the autoregulation capabilities of your body and the biochemistry. And in the coming 3 to 4 years, the organs and their functions will get impaired, cellular physiological changes as well as chronic degeneration will occur. And on top of that, homotoxins from the environment, your food, your water, the air, the soil, heavy metal poisoning, electrosmog, they will all be able to penetrate easier into your body and disturb the cellular polarity and the cell communication even further. Psycho-emotional as well as epigenetic factors together with those homotoxin signals bring then the body out of homeostasis or out of balance, and the autoregulation does not function properly. As a result, the cells will lose their polarity, the membrane potential will decrease, and you will become chronically sick. And this graph shows you very nicely how quickly, within months, unresolved stress situations can lead to this cascade that we have discussed, that eventually leads to chronic diseases such as cancer or diabetes. Therefore, it is very, very crucial that you also pay attention to your psycho-emotional stress situations and resolve them as quickly as possible. Also important to understand when it comes to chronic diseases such as cancer is that it is a so-called morbidic state. And unfortunately, we start to think about our health when it is already too late, when we already have a baggage of several chronic diseases or morbid states, or when we have received such a shocking diagnosis as cancer. A morbid state means that you have several chronic conditions that manifest at the same time, and different organs and their functions are out of balance or out of homeostasis. It is a mistake to treat therefore only the tumor and the organ that is directly affected. In a morbid state, there is a chronic autonomic nervous system dysregulation, an unbalanced biochemistry, so your hormones and enzymes out of balance, and a so-called acidosis, but also cell metabolism that is distortional and thereby creates starvation, toxicity, and cellular hypoxia. So we need to address all of these points if we want to address cancer. Another point that we have to pay attention to when we like to address cancer is the so-called cellular depolarization that makes cancer grow. What do we mean by that? Well, the human body is energy and information that is manifested in a condensed form, which we perceive as our physical bodies. And in order to stay alive and healthy, the energy must flow unhindered through our whole bodies. And therefore, healthy cells have a bipolar structure and they work like a battery. Life energy, also known as ki energy or prana energy, is freely flowing through these bipolar cells when they are healthy. And the positive pole is the top of the body that includes the visceral organs and the left side of the body. And the negative pole is the bottom of the body, so the limbs and the right side. Maintaining this bipolar structure allows the cells to permanently create their living energy that they need, and sick cells become nonpolar, so either plus plus or minus minus, and this neither lets the energy flow through correctly, or it doesn't let the cells create the energy that they need. They are instead consuming energy from the other cells up to 800 times more than they would normally do. And all healthy cells also communicate among themselves, and the sick cells become isolated and cannot communicate or be seen by your immune system. And from this graph you can see very well the difference between a normal cell that is healthy and working properly and a cancer cell that has become non-polar. On top you have the A type, so the normal stem cell, and how it works. It is in true homeostasis, and then on the bottom on the left we have the cancer cell that becomes abnormal or non-polar, and the results of it are the mutations, the tumors that are developing. The other thing that happens within our bodies is that the connective tissue becomes dysregulated as well. So diseases of all kinds, as cancer as well, start always in the extracellular space known as the connective tissue. And most of the toxins are stored in this collagen, elastin, cartilages, the fascia, your fat tissue, the ligaments, and all of those together are known as the so-called matrix, the connective tissue matrix. And the interface between the cells and the connective tissue is the so-called cellular membrane. This membrane is responsible for nutrient penetration to nourish the cells and for cellular waste excretion. And if the cellular membrane has lost its permeability and the electrical potential is significantly reduced or even lost, you will become sick as a result. The inside of the cells and as well on the outside of the cells in the extracellular space there is water, and the total water content in the human body is more than 75%. Thus water plays a very important role in our bodies. And there are three other very important parameters that favorize cancer growth. Chronic diseases as well as degenerative processes, they all develop in an acidic milieu. And three parameters are of importance here: the pH value, the so-called RH2 value, and R for blood, urine, and saliva. pH values determine the acidity and alkalinity of the body, and ideally the blood pH should be between 7.35 and 7.45, the saliva pH at 6.5, and urine pH varies during the day, however there is an average of about 6.8. RH2, also known as redox potential, shows the electron surplus at a specific pH value and the value of the oxygen-hydrogen partial pressure on a scale from 0 to 42, with a neutral value at 28, where the partial pressure of hydrogen is equal to that of oxygen. Biologically ideal, the values for the RH2 should be between 22 and 24 for all of the fluids, including your blood and saliva. And the third value is the electrical conductivity, the R value. And this is a result of electrolytes, meaning minerals in your body. Another factor in cancer development and other chronic diseases actually that is unfortunately not often discussed is the microparasitic infection that can be observed in all cancer patients. And this was already shown during the period of 1965 to 1990 by Dr. Alfons Weber and many other renowned biologists, doctors, and renowned R&D institutes such as the Pettenkofer Institute, the Koch Institute, and the Pasteur Institute that developed and experimentally, undoubtedly demonstrated that cancer is a microparasitic, so a protozoan infection that starts in your red blood cells. Dr. Weber's work is based on thousands of biological in vitro and in vivo experiments with tissues and blood samples collected from cancer patients during their life and even after their death. And as you will see also in a short video sample from his video microscopy, all blood and tissue tests confirmed that microparasites were present in the blood, plasma, and the cancerous tissue of those patients. The cancer theory that it is a microparasitic infection that causes it is exceptionally well and accurately documented, and Dr. Alfons Weber filmed live hundreds of his tests and blood samples measurements that he did throughout the different cancer stages of those patients. And I'm going to show you just a very short sample of those soon. Microparasites as the single real cause of cancer, and this, by the way, this theory is widely supported by many studies and experiments that have been performed, as I've mentioned, by other institutes as well, not only by Dr. Weber, so it was 100% reproducible what he showed. And today, even parts of the Medicine University in Munich, Germany, and also Dr. Alan Cantwell, Professor Dr. Peter Duisburg, Sir Henry Britlin, Wilhelm Reich, and others have all confirmed this theory. And conclusions to all of these studies are that the protozoa are more pathogenic than any bacteria or fungi taken together, and they are the real cause of cancer and many other chronic diseases. Which are the characteristics of those microparasites? Well, protozoa are not viruses, bacteria, or fungus species. They are one single cell living organisms that they were the first life form that developed on this planet, as I've already mentioned. And we can say that microparasites are therefore the beginning of life on Earth. Therefore, these minuscule animals are very resilient and well adapted. They can independently move around and may have even a suction cup organ. They have their own metabolism. They use the same gases as animals and humans, so oxygen and nitrogen. They feed themselves with glucose and proteins and use water and other fluids. They also absorb light and sound. They are so-called unicellular organisms with a cellular membrane. They also spray many toxic substances in their host cells and around in their connective tissue of the host. With isolated protozoa, you can also induce cancer in animals and humans, opposite to tumor cells cloning, which does not produce a cancerous tumor. Protozoa can also be grown in fertilized chicken eggs, and they proliferate through so-called mitosis as well as asexual amitosis at a very high rate. And they use the human body cell's energy, and thus the membrane potential will decrease from 70 to 100 millivolt, which is normal for a healthy cell, down to 15 and even lower. Protozoas also range in diameter from a few thousandths of a millimeter to several millimeters. And you will see clearly that your organism is infested by those microparasites, and your defense systems cannot deal with this invasion anymore because it becomes overwhelming through the many symptoms. First, your blood quality changes. So the viscosity increases, the erythrocytes clump and lose their natural biconcave shape, and the cell's superficial tension decreases due to the changes in the electrical charge that we've discussed, and the oxygen transport is severely hampered, which leads to cell death. Also, blood circulation is significantly impaired because the infected erythrocytes lose hemoglobin, so they go into anemic state, and so the glucose, the plasma, the protein serum, and the cholesterol, and all of these substances will be now missing in your metabolism. And this is why cancer patients have also very often diabetes 2, anemia, or even leukemia, high cholesterol values, as well as globulin values. They are also very often cyanotic, undernourished, tired, fatigued, and their whole system is toxic. Also observable is so-called hyperplasia, which is boosted to enhance the stem cell division and proliferation, which will then be followed soon by so-called hypoplasia, and leukemia being one of the consequences of a blood hypoplasia. Erythrocytes and lymphocytes counts will also diminish, so dead cells and cellular debris will be swimming in the bloodstream. The capillary permeability will be reduced, and general capillary circulation as well will be impaired more and more because the gases cannot be purged, the oxygen cannot permeate into the capillaries, and blood will become under-oxygenated, and also cellular debris cannot be excreted, and that's why patients with cancer are very often in the toxic state. Also, the infected blood plasma will be leaking out into the connective tissue, so we go extravascular and into the organs, and the protozoa now can infect also the lymphatic vessels and the lymph nodes, but also the endocrine glands, the cartilages, and your organs, and this is stage 2 or 3 of the extravascular compensated. Also, the blood vessels will become either too dilated or too constricted or obstructed. Some will become even completely blocked. Aneurysms may appear to support blood circulation. Other side effects of these plasmodial infections are microinfections, necrosis, obturations, chronic heart insufficiency, heart arrhythmia, valve insufficiency, and this may lead even to heart attacks or strokes. Also, the microparasites invasion into the lymphatic system will lead to swollen and painful lymph nodes. So how do the stem cells proliferate and create tumors when they are infected with those microparasites? Well, the mutated cells are the result of the microparasitical invasion of the stem cells. And in order to get rid of the overwhelmingly invading protozoa, the stem cells start to overproliferate in order to dilute the number of invaders per stem cell. The parasites that have invaded the stem cells, as a result of this invasion, they lose the correct genetic information and mutations start to occur as a result. And when foreign parasitical DNA material interferes with our own human DNA, the proliferation and cellular division is potentiated with the aim of saving their own DNA and discarding the invader's DNA. When it comes to spontaneous cancer remission, it's actually a longer process than it was initially considered to be, and is triggered when the parasitic infection was completely healed. Dr. Weber demonstrated that disturbances of mitosis in cancer cases are actually not endogenic and genetically controlled, but exogenic, so based on infectious degree of the hosting organism. And cells may also divide, forming new multiple nuclei cells, like, for example, in leprosy or syphilis. Also, when the body is depleted of its nutrients, mitosis abruptly stops and the cells are going to die. And the brain, blood, and liver are the most resistant organs when it comes to this, and the kidneys and our lungs, the most exposed ones. And the first cancer manifestations after the protozoa have gone through the 4 stages of evolution. They will appear on the skin through exanthemas, rashes, eczemas, and problems with skin-dependent organs such as mucosa membrane, the epithelial layer, also hair, nails. You will see acne, rosacea, psoriasis, scleroderma, syphilis, skin rash, etc. At stage 2 and 3 of the parasitic infection, you might see fever and night sweats, whereby the sweats are outside and inside of the body, affecting dramatically different organs, which now become damaged as a result. Dr. Weber also found protozoa in all patients with polyarthritis, osteoporosis, rheumatism, kidney and liver disorders, lung conditions, dermatologic conditions, as well as psychological and mental conditions. So which are the infection stages that we go through with this microparasitic infection? The microparasites first penetrate the red blood cells, the so-called erythrocytes, where they proliferate and multiply. And then they practically break and dissolve the capillary blood vessel walls using their lysosome enzymes. At this stage, the parasites invasively migrate extravascularly into your connective tissue matrix and your organ's parenchyma, where the organ's tissue will become destroyed and where it creates what we know today as a so-called degenerative chronic disease. A final stage of this Yay protozoa infection is marked by the invasion of your stem cells, and the first defense mechanism of the stem cells is so-called vacuolization, so the enclosure of the parasites into small blisters. And when there are too many vacuoles, the stem cells start dividing in order to minimize the number of vacuoles that are stuffed with parasites within them, and this is known as mitosis and leads to hyperplasia. Once the nutrients for the parasites that are kept enclosed in the stem cell vacuoles have dried up, the stem cells start dividing in order to reduce the number of parasites per cell, or there is a stem cell apoptosis, so a cell death, due to the toxicity that has been created within the cells and the starvation. At this stage, the microparasites become free again, and depending on the organs that are invaded and the patient's general state of health, the first symptoms appear in form of skin rashes, skin eczemas, joint and lymphatic system inflammations and edemas, also dysfunctions, and up to or total shutdown of the organ's functionality. And the cancer tumor is the latest stage of this infectious process that develops in 4 stages. First, the protozoa intravascular compensated infections, so they are in the blood vessels. And then the protozoa intravascular decompensated infection, which also still takes place in your blood vessels. The third stage is the protozoa extravascular compensated infection, where the protozoa migrate from your blood vessels into your connective tissue and organs. And the fourth stage is the so-called protozoa extravascular decompensated infection, where they migrate into your stem cells. And the cancer stages through the protozoa infection are very similar actually to the malaria stages or the toxoplasmosis infection that also develops most likely therefore from this protozoa class. The microparasites described by Dr. Weber that you can see here can be found in the tissue of all cancer patients. Some examples. Here you can see a blood sample that has been dried over a flame. The patient suffers from leukemia. Here you see the blood sample of a patient suffering from liver cancer. Here the blood of a patient with bladder cancer. And this is tissue from the bladder carcinoma. You can clearly see the lively movements of the microbes. Here the diagnosis is that of lymph node metastasis. This is an almost mature gamete. He can be found in the space between the cells. And here a blood sample after mammal surgery. You can see a dying leukocyte. Patient is suffering from breast cancer. Diagnosis: cell complex of an uterus tumor. This is the blood of a patient suffering from a lip carcinoma. And here cells of a lung carcinoma. In the lower left part you can clearly see red blood cells in whose innards there are lively microparasites. Here a tumor cell from a bladder carcinoma. You can clearly see the vacuoles. String-shaped structures. All recordings have one thing in common. You can see certain reoccurring types of microbes in all of them. There is no tumor tissue without these microbes. There is no blood of a cancer patient without those microparasites. Experiment A. Thick drop of blood, temperature 37 degrees Celsius. We take a blood sample from the patient and follow Dr. Weber's instructions to prove his theory. With the capillary blood we prepare a native blood spread. We pass the sample through the flame 3 to 4 times. The temperature reaches 140 to 160 degrees Celsius. The blood cells are now cooked and totally dried up. They are definitely dead with all of the existing organelles and vesicles. Then leave the sample to cool down to body temperature. We now use a sterile 1 ml syringe and fill it with 0.3 ml of natrium citrate solution, Fresenius, and 0.7 ml of sterile Ringer solution, Fresenius. We mix the contents of the syringe well by shaking it. We now drip a bigger drop of this solution onto our sample and cover our sample afterwards. We look at the sample through the microscope in an oil emulsion at an aperture of 2 to 2.5. The destroyed blood cells can be seen very nicely without any movement. After a short period of time, something starts to happen, however. The parasites inside of the dead blood cells come back to life. We can see them swimming around vividly and very actively. They are self-sufficient life forms and not apoptotic bodies. All human cell material is 100% dead in this sample. Experiment B is the same procedure as with A, but this time we use a very thin blood spread. Dr. Weber claims that so-called apoptotic bodies are independent life forms. To prove this, we destroy freshly taken blood by heating it up to 140 to 160 degrees Celsius again. But the parasitical microbes proved to be almost indestructible. Despite the heat, they remained unharmed and very alive, as you can see. Under the microscope, we can observe similar results to the ones in experiment A. We can see again the remains of the dead red blood cells and no movement. After the application of the Ringer solution, we can see again parasitical microbes that are clearly independent life forms. Alright, so with all that in mind, let's talk about what a correct cancer therapy that really works has to include in order for it to be effective. Well, first, the cancer cells, as we've learned, are always non-polar. So they are just the opposite of healthy cells, which are dipose battery-like. So the correct maintenance and the cell polarity is required for the development and homeostasis of all metazones. Cell polarity mechanisms are also responsible for the regulation of the asymmetric cell divisions of the stem cells, and the disruption of the cell polarity is a hallmark of cancer. Furthermore, recent evidence indicates that the loss of the cell polarity is intimately involved in cancer. Secondly, the cancer cells thrive through fermentation of glucose. The metabolism of primitive unicellular protozoa when they have adapted from aquatic life and the lack of phosphorylation pathway makes cancer cells crave glucose, and the lack of their energy is compensated by stealing up to 800 times the energy from the healthy cells. Also, the genetic mutations are due to a DNA exchange between the protozoa and the human stem cells during the cancer tumor proliferation, as we have seen. You are not born with genetic mutations and a predisposition to cancer, therefore. Also, there is always a protozoa reinfection risk. Microparasites do not create immunity. So first and foremost, therefore, the first action in cancer therapy must be a repolarization of the sick cells, and this also implies an increase of the cellular membrane potential to 70 millivolts or higher. This requires a lot of electrons to be made available. Also, we need to stop the fermentation by sugar starving the organisms and re-establishing a 36 ATP metabolism based on glycolysis and oxidative phosphorylation. So we need to correct and improve the mitochondria respiration. Thirdly, we need to stop the development of cancer from a microparasitic intravascular infection in the blood vessels to a protozoa extravascular migration from the blood vessels into your connective tissue, and finally to a protozoa extravascular decompensated infection by invasion of those protozoa into your stem cells. And the cancer development as a protozoa infection cycle is very similar to a malaria infection or toxoplasmosis. And also, when the microparasitical infection has been eradicated, the genetic mutations can be reversed through a healthy lifestyle and regular matrix regeneration therapy. So we need to do this as well. We also need, of course, to adopt a healthy lifestyle and regularly follow detoxification and disinfection procedures to prevent reinfection and further development of the microparasites. We need to avoid biopsies and surgeries if possible, because this only helps the protozoa spread even faster. And chemotherapy might work, but is always at the cost of our general health. And how we will do this specifically, with which devices and natural supplements as well as natural remedies, all of this will be discussed in the second part of this presentation that is going to be out soon. So subscribe and also hit the notifications button to not miss out on this very important part. If you have any questions about what we've discussed in this presentation, do not hesitate to contact us. shop@lyranara.com. I'm also going to post our full contact information, including our toll-free phone number, in the description box below for you. If you've enjoyed this, also like, share, and of course, as I've mentioned already, subscribe to support our work. It's very important. And until next time, stay healthy.